Okay, now we're moving on to question I through K, and this is the epistasis problem. So if we go back to our original wild type and mutant situations, where we have the wild type with some position effect variegation, some white patches, some brown patches, we have our two mutant phenotypes. We have the SUVARs, where we have the suppressor variegation, so that means that our eye color is going to be more brown. We have our EVARs, which are the enhancers of variegation, which means that our eyes are now going to be more white. So if we know the loss of function phenotype for these mutants, we can then infer the wild type functions of the genes. So if the SUVARs have less silencing than in the mutant, then you would infer that the wild type function of SUVAR is to promote heterochromatin formation. And then by contrast, the converse would be true for EVAR, so that the wild type function of EVARs is actually to inhibit heterochromatin formation. So now I want to do the epistasis analysis. When we have two mutant phenotypes that are opposite of one another, then we can do an epistatic relationship in a sort of a binary pathway or biosynthetic, a binary or a regulatory type of pathway. So here we have our single and our double mutant phenotypes. And so suppressor 2 has brown eyes, enhancer 1 has white eyes, enhancer 2 has white eyes. Okay, so those are two opposite phenotypes that we can then make the double mutants and see, do we get white eyes or do we get brown eyes? And so here, if we make the double mutant for either S2 and E1 or S2 and E2, in both cases, the double mutant has a white phenotype. That means they have the phenotype of the enhancers. The enhancers, so by knowing that the E1 and E2 have, that these have the phenotype of E1 and E2, that means that E1 and E2 are epistatic to the suppressor phenotype. So E1 and E2 are epistatic to S2. So that we know when there's a negative relationship between the genes in a genetic pathway, that the epistatic allele defines the gene that functions downstream in the pathway. So that would mean, and they have a negative relationship, so this means that S2 is upstream of E1, and the relationship to each other, we would put a bar because they have a negative relationship. Now, since we have two EVARs, and we know that they both act in the same, they both have a similar function, if we were to order them in a pathway, we would put an arrow between them because they both contribute to the same effect. So in both cases, we know that E1 and E2 are both downstream of S2. And here we're talking about the wild type functions of these genes. Okay, whereas these, that's a mutant phenotype. So if we were, so we know that E1 and E2 are both downstream of S2, and there's this negative relationship, but we know that the relationship between E1 and E2 is a positive relationship. Okay, so if there's a positive relationship, they should have an arrow between them. But the two of them, each one of them is an inhibitor of heterochromatin. So we would draw the pathway as follows. We don't know the order of E1 and E2 with respect to each other. They have the same phenotype. But if we ordered them in a pathway, we could either put E1 going to E2 or E2 going to E1. And so that is the answer to the last part of that question, which is draw two possible pathways that could involve all three of these genes.